Hello, we are Group 5. We're presenting on tobacco and cancer. My name is Danielle Alvarado.
My name is Kaylee Marquardt.
My name is Shantae Moore.
And my name is Jason Rodriguez.
Today we'll be going over 5 different topics. First, cancer and tobacco, then current knowledge of the field. After that, research findings and a summary of our main points as well as our references. First, we'll be discussing understanding the link between cancer and tobacco.
Okay, the link between tobacco and cancer. Tobacco remains one of the biggest causes of cancer that we can actually do something about. Most people think of lung cancer when they hear about smoking, but in reality, tobacco use is tied to many different types of cancer. Tobacco refers to products created from the plant Nicotiana, which are used through forms of smoking, but also chewing, such as cigarettes, cigars, chewing tobacco, etc. The use of tobacco is the #1 preventable cause of cancer worldwide. According to the World Health Organization, or WHO, 25% of deaths by cancer around the world are caused by the use of tobacco, and is the primary cause of lung cancer, which make up 1.8 million deaths per year, and 18% of deaths of all cancer deaths in the world. When it comes to statistics on tobacco related cancer, in the United States, approximately 660,000 people are diagnosed with a cancer due to tobacco, with 343,000 a year dying from these cancers. Tobacco is also the primary cause of lung cancer, and it is linked to mouth, throat, esophagus, pancreas, bladder, liver, kidney, and cervix cancers. Tobacco is also linked to leukemia cancers. Secondhand smoking causes 7,300 lung cancer deaths a year to people who do not even smoke. This represents how tobacco affects more than just those who use it. What's even more concerning is how unevenly this impact is spread. People in lower income communities or those with less access to education and healthcare tend to be affected the most, as represented on the next slide. Adults from 45 to 64 years old have the highest rate of cigarette smoking. When it comes to geographic location, smoking rates are highest in the Northeast, with 202 people smoking for every 100,000 people, and lowest in the West, with 170 for every 100,000 people. Higher rates of smoking are usually seen in locations with lower education and higher poverty. This represents the fact that in the Northeast, there are lower education and higher poverty rates, whereas in the West, there is a higher education and lower poverty rate. Men have a higher chance of being diagnosed with cancer than women, with 250 men being diagnosed for every 100,000 men, and 148 women out of every 100,000 women. This represents the fact that there are many more men who smoke than women, and many more men who use tobacco versus women. All of this highlights how important it is not to, not just to raise awareness, but to also support resources to quit and actually reaching people who need it the most. Reducing tobacco use could literally save hundreds of thousands of lives.
This figure displays the link between smoking tobacco and cancer. Tobacco or nicotine is exposed and absorbed into the bloodstream. Tobacco has more than 2,500 chemical components, with many being known as human carcinogens. Once the carcinogens are absorbed into and transported through the bloodstream, it goes through metabolic activation and creates DNA adducts. Some of the carcinogens that are majorly involved in this process are polycyclic aromatic hydrocarbons. And shortened, it's called PAOS. N-nitroamines, such as NNK and nicotine. DNA adducts are formed through carcinogens in tobacco, PAA in particular being benzoapyrenine, aka BAP. BAP metabolizes into a more reactive form and adheres to DNA to form DNA adducts. From the DNA adducts, if there is a persistence in miscoding in the DNA, it can lead to mutations in oncogenes and tumor suppressor genes. According to Rong-Jane Chen et al., adduct formation is most usually found at adenine or guanine sites of DNA. These adduct formations can lead to mutations such as KRAS oncogene in lung cancer or the TP53 gene in several other cancers. Mutations can activate oncogene genes, and mutations in DNA can inactivate tumor suppressor genes, which can lead to uncontrolled cell proliferation. The genes that are mainly affected by tobacco and assist in the production of cancer cells are TP53, KRAS, and EGFR. DNA methylation histone modifications are some epigenetic changes that can occur from smoking tobacco through changing the gene expression. The carcinogens in tobacco can alter histone modifications that will cause for the activation of oncogene genes and the inactivation of tumor suppressor genes. The top pathway displays that after the initiation or uptake of carcinogens, there is receptor binding, then protein kinase A and B activation, which leads to a loss of normal growth control mechanisms and causes cancer development. The bottom pathway displays that after the initiation of cigarette smoking, that there is an uptake of co-carcinogens and tumor promoters that leads to tumor suppressor gene inactivation and other changes that subsequently leads to the loss of normal growth control mechanisms and causes cancer development. The specific types of cancer generally depend on which organs and tissues are exposed to the tobacco smoke, in which the lungs are amongst the most impacted. In the next slide, in carcinogenesis, there are 3 stages known as initiation, promotion, and progression. In this figure, nitrosamines and PAOs are absorbed into normal cells and create genotoxic effects that lead to the initiation stage of cancer. The enzymes are activated and ROS production occurs. ROS production can also be associated with inflammation and oxidative stress. As previously stated, PAOs such as BAP induce the formation of DNA adducts, and if the DNA cannot be repaired, then this leads to the initiation of cancer. According to Rong-Jane Chen et al., the continuous exposure of ROS can lead to the mitochondrial DNA mutation. These mutations have a faster growth rate and are considered aggressive tumor growths because of the oxidative damage smoking tobacco causes. TP53 is an important tumor suppressor gene in the cell cycle. It is known for cell cycle arrest, repairing DNA, and inducing apoptosis. The mutations that inactivate TP53 allow for cells with damaged DNA to continue throughout the cell cycle and continually divide. This leads to the promotion of tumorigenesis and the continual gathering of mutations. Retinoblastoma is another tumor suppressor protein that is affected by genetic mutations, as it is a protein that makes sure the damaged DNA in the cell would not pass through the G1 phase, going into the S phase. It goes through this function by binding and inhibiting E2F transcription factors. When retinoblastoma is altered and loses its functions, it allows for the cell cycle to continually progress even if there is damaged DNA present, which in turn causes the progression of cancer. Mutations in TP53 are prevalent in tobacco-related cancers in the lungs, oral cavity, neck, and head, and mutations in retinoblastoma are prevalent in the lungs, neck, and head. In the next slide, in this figure, it shows the non-genotoxic effects that nicotine, nitrosamines, and PEAs have on initiated cells and tumor cells. These effects activate receptors such as nicotinic acetylcholine receptors, beta adrenoceptors, and aryl hydrocarbon receptors. One of the subtypes of nicotinic acetylcholine receptors, also known as alpha-7 nicotinic acetylcholine receptors, is activated by nicotine and is involved in angiogenesis through causing inflammation, ischemia, and supplying tumors with blood vessels. Overexpression or activation of these three receptors may cause further release of neurotransmitters and growth factors that partake in angiogenesis, invasion, metastasis, cell proliferation, and the inhibition of cell death. pKa, 5-LOX, STAT3, and PPAR beta and delta are activated and prevalent in the promotion cancer stage, while the signaling pathways and molecules that are involved in both cancer promotion and progression through tobacco are PKC, PI3K, AKT, MAPK, ERK, and COX2. The PI3K AKT pathway regulates cell survival, growth, and metabolism. Cellular proliferation from this pathway is increased when NNK is present, as NNK binds to nicotinic acetylcholine receptors to induce cell proliferation, survival, and tumorigenesis. It is also suggested that nicotine and NNK can activate different signaling molecules and pathways such as ERK1 and 2, STAT3, or AKT to obstruct anti-growth cells to increase cancer cell promotion and progression. The MAPK-ERK pathway is regulating cell differentiation, proliferation, and survival. According to Rong, Jane, Chen et al., nicotine and NNK bring about the transformation of human bronchial epithelial cells into malignant cells through inducing the survivin mRNA expression through this pathway and in the survivin protein found in lung epithelial cells. VEGF promotes angiogenesis. COX2 and EGFR are important in cell proliferation and transformation. The activation of PKC and SRC caused by nicotine increases angiogenesis and invasion in the progression stage of cancer. Nitric oxide is a vasodilator, and when nitric oxide production is increased, it can stimulate angiogenesis. And nicotine promotes metastasis and progression of cancer through the increased expression of chemokines as well. Chemokines such as CXCR4 and CXCL12 are the key factors for tumor migration metastasis. Overall, the interactions of several pathways that include the damage made to DNA, the activation of oncogenes, the suppression of tumor suppression genes, and genotoxic and non-genotoxic effects, along with the inflammation and the suppression of the immune system, make up the three stages of the development in cancer, such as initiation, promotion, and progression.
Now that we've gone over some statistics on tobacco and cancer and the molecular mechanisms involved in cancer related to tobacco use, we'll delve into current knowledge on the topic. So as mentioned before, we know that different cancers can develop due to the use of tobacco products like smoking cigarettes. And most of us think about tobacco-related cancers as being only in the lung. However, these cancers can develop all throughout the body, including throughout the esophagus. It can result in leukemia, stomach, pancreatic, and even cervical cancer. However, as mentioned before, lung cancer is the most prevalent cancer that is related to tobacco use, with around 90% of lung cancer related deaths being linked to tobacco use. We can also see that cancer mortality rates do increase as intensity of smoking increases. So in this figure from a paper published in 2002 by Cupper, we can see that compared to non-smokers, those that smoke between 1 to 25 plus cigarettes per day have an increased relative risk of developing different types of cancers related to tobacco use. So if we compare in the top graph, the relative risk of those that are non-smokers compared to those that smoke, we can see that non-smokers have a relative risk close to 0% of developing cancer or dying of related cancers, compared to those that smoke 25 or more cigarettes per day, have a higher relative risk of dying of lung cancer or esophageal cancer by 25-fold. Also, if we take a look at the graph at the bottom, we see that non-smokers have a relative risk of about 1% of cancer mortality versus those that smoke 25 plus cigarettes have a higher relative risk of dying from tobacco-related cancers like pancreatic cancer, bladder cancer, stomach cancer, or kidney cancer. We also know that smoking and tobacco use results in the creation of these reactive oxygen species, also known as ROS. And these reactive oxygen species are oxygen-containing molecules that can ultimately damage proteins, lipids, or nucleic acids like DNA or our genes. And so we know from a paper that was published in 2021 that this ROS-induced damage can lead to genetic or epigenetic alterations that contribute to carcinogenesis or the development of tobacco-related cancers. So if we take a look at the figure on the right-hand side, we can see that these reactive oxygen species that are caused by the use of smoking products lead to a lesion on DNA, in this case, a lesion on a guanine base, and that can either lead to repair mechanisms, apoptosis or cellular death, or most importantly, genetic or epigenetic alterations that can upregulate tumor suppressor genes or oncogenes leading to cancer. We've also seen by previous studies that nicotine, known as the active ingredient in tobacco, has been linked to cancer cell growth. So while nicotine is not itself classified as a carcinogen on its own, it can induce cancer cell proliferation by interacting with other chemicals found in tobacco, by enhancing angiogenesis or the creation of new blood vessels from existing blood vessels, as well as by suppressing apoptosis. So in a study that was published in 2007, they found that nicotine was shown to stimulate human lung cell carcinoma by inducing the expression of fibronectin. So in this figure on the right-hand side, we can see that the more days that nicotine is given, fibronectin protein levels actually increase, leading to adherence of these lung carcinoma cells. Fibronectin is an extracellular matrix glycoprotein that adheres to lung carcinoma cells and essentially allows for them to stick together and create the tumorigenesis that we see related to cancer. So fibronectin enhances tumorigenicity by allowing for tumors to grow and also prevents apoptosis in response to chemotherapeutic agents, showing how difficult it could be to treat these different cancers due to these glycoproteins that are present. And now, while we've discussed the use of tobacco and its link to cancer risk and mortality, there have been studies that show that quitting tobacco is also linked to reduced cancer risk and mortality. So, in a study that was published by Thompson et al. In 2021, they found that individuals that quit smoking earlier in their lives actually experience lowered risks of dying from tobacco-related cancers. And so on the left-hand side in graph A, we can see that those that start smoking later in life, their cancer deaths are lowered in due to smoking. But when these groups, for example, those that started smoking at ages 18 through 20, when they quit smoking at those same periods, their excess risk of cancer mortality is actually lowered by 100%. There's also been studies that have shown that there are sex-based differences, like have been discussed before, that have been identified between cancer mortality and tobacco use. So a study that was done by Thun et al. In 2002 found that men have a higher probability of cancer as probability of death as current smokers compared to women by almost twice the number of death. And then when we compare those that have never smoked or quit earlier in their life, we can see, like in the previous slide, that their probability of dying of tobacco-related cancers is a lot lower. Now we'll move on to discussing some prominent research findings in this field.
So the first research finding was how nicotine promotes tumor growth and metastasis in mouse models. So the background of this research was basically saying that, nicotine is an addictive compound, which we all know. Nicotine has, so nicotine is found in all tobacco products, but tobacco is not found in all nicotine products. And nicotine also induces cell proliferation and angiogenesis. So, angiogenesis was mentioned before by my other group members. So, angiogenesis is the process by which new blood cells form from existing ones. So, in that process, a signal is sent, blood vessels respond, new vessels grow, and then blood flows in. And in this process, cancer basically hijacks that process from angiogenesis in order to proliferate. So the hypothesis was nicotine may enhance growth and spread of pre-existing tumors. And on the next slide, we show the methods and delivery. So they used two different mice. They used the line one syngenic mice, and then the second one is a carcinogenic mice. So the first one was to assess. They used the methods were sorry, the delivery methods were the microderm patch and the intraperitoneal injection. So the first one, the syngenic mice, was to assess nicotine's impact on growth and reoccurrence after surgery and lung metastasis. So with the Nicoderm patch, it's a 14 milligram patch. They cut them into 30 pieces, and the amount was 25 milligrams per kilogram daily for 2 weeks. And then the intraperitoneal was 1 milligram per kilogram 3 times per week. So the carcinogenic mice, they were used to evaluate the nicotine effect on the number and size of the tumors. So that one was administered the same amount as the first one, of course. So intraperitoneal injection, 1 milligram per kilogram, 3 times per week. And on the next slide, we show the findings. So this is just a graph showing like the nicotine, that nicotine promotes the growth of L1 cells. So if you look at the two graphs to the right, vehicle is basically the control. And then the nicotine, you can see that the levels in the tumor growth are way higher for nicotine than it is for the control. And this was in the study done by Davis and his group members. So the next slide, the summary of that was tumor size increased significantly with nicotine. The reoccurrence rate with nicotine was 59.7%, whereas the vehicle or the control was 19.5%. In lung metastasis, it was 9 times higher in the group with nicotine. On the next slide, we have another research finding, the second one. So this one was for tobacco use and response to immune checkpoint inhibitor therapy in non-small cell lung cancer. So the background of this one is we know that tobacco is a known risk factor for lung cancer. So continued use is linked to less favorable outcomes during surgery, chemotherapy, and radiation. So basically, the more you smoke, the less chance you have of like surviving these therapies or surgeries or the actual therapy working. So the objective was to review the association between tobacco use and immune checkpoint therapy. So, immune checkpoint therapy is, I'm not sure if we went over that before, but the checkpoint is basically a break that slows immune cells. And the checkpoints are usually PD-1 or PDL1. And when active, they stop T cells. So the ICI or immune checkpoint inhibitor drugs, they block these breaks so T cells can fight cancer. So this review was to analyze phase 3 clinical trial data comparing smokers versus non or never smokers. And on the next slide, we can go over the methods. So for this one, it was more a review. So the methods that were used were literature. So for literature, they reviewed existing literature, examining association between tobacco use and tumor biology in the context of immunotherapy. They also use hazard ratios, which is a statistical measure to compare the outcomes between current smokers and ex-smokers, as well as never smokers. They also used subgroup analysis, which is based on smoking history, the comparison of efficacy on the ICI therapy between ex-smokers and current smokers. This includes treatment of naive setting and then pre-treated settings, and also ex-smokers versus current smokers. And on the next slide, this is a visual. So basically, in this visual, this was from the review written by Cork. So in this one, it basically explains that how smoking changes the immune environment in lung cancer. So tobacco smoking basically weakens the body's ability to fight tumors by overactivating inflammation, boosting the function of regulatory T cells, which suppress immune responses, as well as lowering the killing power of natural killer cells, reducing B cell activity and the production of antibodies. So like the IgA, IgG, and IgM, and as well as suppressing the development and activity of dendritic cells, which help alert the immune system. So this is just showing how the carcinogens go through the process of weakening the immune system or in the lungs and causing cancer or contributing to cancer. So the next slide, the summary of this literature review was basically that smokers showed better response to ICIs than never smokers. And then ex-smokers benefit more than current smokers. And we can go to the next slide. So here we'll go over some of the summary of our main points. So I'll go over the first one. The use of tobacco has been linked to the development of cancers, particularly lung cancer. Tobacco significantly affects chance of diagnosis of cancers and cause 90% of lung cancer deaths. And I'll let my group members go over the next two.
So to take it off, older aged adults, like we had mentioned, men, as well as those that are less educated and live in populations that experience poverty, are often at higher risks for tobacco-related cancers than others. And while we know that there are many components in tobacco, the three main components that are studied the most are nicotine, nicotine-derived nitrosamine ketones, or NNK, and polycyclic aromatic hydrocarbons, or PHAs. As well as we covered, the overexpression or activation of receptors can ultimately lead to cellular proliferation, angiogenesis, or the creation of new blood vessels, metastasis, invasion, and the inhibition of cell death. We also discussed that cancer mortality rates and cancer cell growth are positively correlated with the use of tobacco or other smoking products that contain nicotine. Tobacco use also induces the creation of these reactive oxygen species that ultimately lead to genetic and epigenetic changes in cancer. And ultimately, quitting tobacco can reduce cancer risk and mortality, which also shows sex-dependent characteristics. This undermines the need for having programs that allow individuals to quit tobacco or nicotine use in order to decrease lung cancers or other cancers that are associated with tobacco use and their mortality. Now we'll go over some of our references. As we can see here, these are the references we used for the papers and the peer-reviewed studies. And thank you so much. We hope you enjoyed.